Howdy, folks. Welcome back to Bio 5. So this week we're going to be looking at evolution and ecology. I've done some modification. I looked at I want to go and remind everyone that this is going to be the last week of content before we go into finals. So this is the last bit of content before we go into finals. So, zone in, make sure you're focusing. You guys got this. I have a lot of confidence in you. Everything you're gonna need is going to go and be on the website. It is worth noting that since this recording is going to be reused in the future by other teachers and such, I'm not going to go say the dates and stuff, but I will post announcements on the Canvas. Alrighty. With that, I think we should get started. So let's start with chapter 22, Evolution and Our Heritage. Alrighty. Now I drag you over here. Slideshow from the beginning. There we go. So we're going to be talking about evolution and our heritage. So let's go over the outline for this. So evolution of life on Earth. We're going to cover the evolution of life on Earth. We're going to talk about the scale of evolutionary change. We're going to talk about some of the evidence, the support evolution, and we're going to go and talk about how evolution has affected humanity. So evolution can be summarized as descent with modification. The way that it's going to work, so you're going to have an organism, and they are going to have subtle changes to their DNA structure, and then through the process of sexual reproduction, which we covered last week, their genetic data is going to be recombined with another individual, and then that offspring is going to have certain traits it inherited from either parent. If it inherited traits that are going to allow it to go and have more offspring that make it to reproductive age, then that trait's going to be what we call selected for. So, yeah, there is no sapience to this. It is just statistics. But if a trait is inherited that helps keeps an animal alive, they're going to live longer to reproduce, and because they're going to live longer to reproduce, they're more likely to pass that trait onward. So that's what this is talking about. Now, life on Earth is estimated to be about 4.5 billion years old. This number since this time, I think, has been pushed up to about 5 billion. Now, the environment, very, very different from that of today. Super hot volcanic crust 4.5 billion years ago. Incredibly intense UV radiation and almost no gaseous oxygen. The atmosphere at this point was mainly nitrogen and CO2. Now, so as the planet is 4.5 billion years old, we have evidence for life for up to 3.8 billion years ago. It is possible life has existed earlier than this, but we do not currently have evidence to suggest so. This is roughly what it looked like. A lot of lava flows, a lot of lightning, pretty much constant chemical rainstorms. The only difference here is the atmosphere should be kind of reddish because of the high proportion of um, greenhouse gases that's going to be out here. Now, chemical evolution. So, life evolved from chemicals of steadily increasing complexity over the course of 300 million years. So, basically, we started with like our base genetic material, our base amino acids. Then, after a while, we started forming proteins, and we started conveying information on RNA and proteins, and that eventually allowed for life to emerge. Life is the continuation of a series of blueprints. Your DNA is going to tell your body how to grow. Then you're going to go and grow and reproduce and make a bunch of little babies that are going to be built off of that blueprint. Uh, in theory, drawing a line between a self-perpetuating chemical reaction and life is kind of difficult. So we have some possible steps in chemical evolution. We start with inorganic molecules and small molecules. And then after a while, we started working up to macromolecules. Each of those can be used to go and transmit data between generations in one way or another. After life has evolved, we started developing the first life forms were going to be the early cells. So that would be the prokaryotic or the non right or the non organelle having cells, or eukaryotic, which means it has membrane-bound organelles. Now, a multicellular organism is going to be an organism that's composed of many, many different cells working together. This did not emerge until significantly after the evolution of life. The majority of life on this planet and the history of this planet has been single cellular. Now, small organic molecules, inorganic molecules that form small organic molecules. So that would be things like oxygen and carbon and nitrogen. All those are elemental. They're not technically organic, but all organic molecules are made out of them. Now, energy. Over the course of this process, we are pretty sure that the low oxygen atmosphere, combined with the particular organic molecules that were found in like the primordial sea, combined with energy being provided by lightning and intense UV radiation, starts to make molecules that are the blueprints for life. So this was tested by several scientists, and they started to see the small organic molecules starting to form larger molecules. After a while, proteins and even crude nucleic acids started to form. This is the best thing that we have to work for. This is the best model we have to work with. So this is the experiment that was run by those scientists. And they created a tiny atmosphere, simulated the atmospheric conditions from 3.5 billion years ago. And sorry. Yeah. So we start with the primordial sea. This simulates the water conditions of the primordial sea. Then we set that to a boil. This then precipitates out, into this gas chamber, where the atmospheric conditions of the Triassic Sea were being stimulated, the primordial sea are being simulated. An electrical current was passed through these conditions, and then those products were allowed to precipitate out of the bottom. This precipitant was then caught here. And in it, we found a bunch of newly formed organic molecules, specifically the foundational molecules for the larger macromolecules that would later emerge. Now, macromolecules in early cells. So organic macromolecules, proteins and genetic material. These would then aggregate into droplets, and these were kind of the precursor of the living cell. So these all were prokaryotic single cellular organisms. They originated about 3.8 billion years ago. Then about 1.8 billion years ago, eukaryotic cells started to evolve. So membrane-bound organelles. Now, we are reasonably sure that some of the original organelles are actually descendants of free-living bacteria that were phagocytized by a host cell. Basically, they had a symbiotic relationship, and one of the best pieces of evidence we have for this is that the mitochondria is filled with nuclear material. It has its own DNA that is separate from your DNA. We actually call it the maternal DNA because it is going to go and be strictly genetic lineage that's passed through the female line. And because it's not being reorganized through sexual selection, it's like strangely static, which can allow us to do some pretty interesting genetic testing and go and actually trace human lineage back to a point of like our last common ancestor, which is pretty cool. Now, multicellularity evolved about 0.2 billion years after that, around 1.6 billion years ago. This evolved out of eukaryotes. Organelles are significantly more efficient than not having any organelles, and a sorry, folks. I don't know why I'm so tired. All right, multicellular organism is going to go and be constantly fighting against the square cube law. It's going to be constantly trying to go and fight its surface area to its volume ratio, and it's always going to be losing. So you need more efficient base units to accommodate for the inefficiency that comes from multicellularity. Eventually, these multicellular organisms became organisms. So this is kind of that flow chart of the origin of life on Earth. Inorganic molecules form small organic molecules. Small organic molecules coalesce under UV radiation or under electricity. You end up with larger organic molecules, including RNA or small proteins, that then start to naturally accumulate into droplets. Eventually, you get the right accumulation of droplets, and you kind of can form a prokaryotic cell. From there, you have a living animal. It spreads, it begets, it does what it do, and then it's going to go and turn into one, and then that's going to specialize into a eukaryotic cell. So eukaryotic cells originated because of phagocytosis of prokaryotic cells absorbing other cells, and then those being and then those differentiating and specializing into different organelles. Eukaryotic cells eventually begat eukaryotic organisms or multicellular organisms, which is just a bunch of different specialized cells working together to keep a single organism alive. So microevolution, these are changes at the genetic level that can occur within a single population over the course of a couple generations. These are super small things that are usually not noticeable. It can be something as simple as like, oh, well, uh, grandpa had a white streak in his hair that showed up out of nowhere when he was born. All his descendants have a white streak in their hair. This could be a point mutation, like a single change, a change in one spot on the genetic code. In addition to that, we also have macroevolution. This is going to be what you typically think of when you're thinking of evolution. Large scale, over a long period of time, that will beget large groups of species, usually triggering mass extinction events. This is the evolution on a large scale when it's defining like a population or like a continent. Now, basic terms in microevolution. So at the low, so for evolution, the smallest unit we can really think about is going to go and be the population. This is going to be a group of individuals of the same species that live in the same area and interact with each other. Any population is going to have a gene pool, which is going to be every allele, which is every possible presentation of every gene that is in the population. So this is going to go and kind of be a database of every single allelic combination that currently every single allele that currently exists in the population. And alleles, again, are just different variations of genes. If I have the black hair gene, I can have, or if I have the hair gene, I could have the brown hair gene, the black hair gene, or the blonde gene. All of them are going to be the same gene, but they're going to be different alleles of each other, going to be different variants. Now, microevolution, variation in populations. This is going to be mainly propagated through sexual reproduction. And instead of being driven by mutation, it's going to just shuffle around previously existing alleles in the population, sexual recombination. And it's going to go and hopefully stumble across a combination that's more effective. And this is happening all the time in all populations. And once enough of these accumulate, it does become a macroevolution. So microevolutions are just the start of the landslide. Now, variation in population. So sexual gametes in a sexually reproducing species, are going to be variable due to crossing over and independent assortment. They're going to have and basically because of the way sexual reproduction works, the way that we are exchanging alleles between individuals, we are recombining DNA from multiple individuals, that is going to go and be giving us genetic variation over time. And it also has to do with the actual processes that are going to be occurring during mitosis and meiosis. So this is where we're going to go and have crossing over and independent assortment. Those just being when the chromosomes, two chromosomes will go and exchange a portion of their DNA. And independent assortment claims that during cellular division, chromosomes are randomly assorted between the two daughter cells. So there's not like a specific definable pattern. Now, the other way that variation enters a population is mutation. So that's where new genes and new alleles and it's going to occur in a so typically a mutation is just going to be negative. It's just going to give you cancer. Occasionally, if you have a non-deleterious mutation, it can be passed. It will be can be passed on to your offspring. You know, if they happen to inherit that chromosome. Mutations have a pretty low rate of occurrence because your body has a lot of things in place to stop mutations from getting passed on, and the reproductive cycle also has a lot of things in place to go and keep mutations from getting passed on. But this is where every single trait in change originally came from. Like this is where the foundational DNA came from, was process of mutation, adding and removing things, messing with the process. Now, some processes that lead to microevolution. That would be genetic drift, gene flow, mutation, and natural selection. So let's go and define each of those terms. We'll start with genetic drift. Genetic drift, basically, because all of this is a matter of probability. Independent assortment, it is just 50-50 which daughter cell is going to go and get the chromosome. Sexual, it's complete. It's kind of like because of this, occasionally statistics can do damage to your subject population. If your population is changing, is having a fluctuation just because of chance. Let's say, even though blue and pink hair both are considered equally attractive in this society, pink hair the second generation only has blue hair, and the pink hair gene has gone locally extinct. It wasn't because of a selective pressure; it was just random chance. That is genetic drift. In general, the larger your population is, the less subject you are to randomness, because it just averages out statistically. So you typically don't see genetic drift in very large populations. It's more of an issue once we start to get down to like a couple hundred individuals. Now, genetic drift can go and be influenced by two different variables, or two different effects, and these are the bottleneck effect and the founder effect. So so basically the bottleneck effect is if a population loses a very large amount of its population, it is statistically likely to lose certain alleles. So like from here, I pull out two M&Ms. By chance, they both happen to be blue. That means only the blue gene was passed on to the next generation. The white gene did not make it to the next survive this event. So, what would this actually be representing? Well, here we start with a large population, and then let's say there is a fire or an earthquake, or some individuals get trapped on a boat and sail off to sea. Of the surviving population, we only had two survivors. We lost 50% of our genetic diversity. We don't have any of the white M&Ms anymore. But now we have 100% blue. This is known as a bottleneck event. Because we lost so much population, now we have lost genetic diversity because we just don't have enough people who made it to the next area. This is known as a bottleneck event, and this population is known as a founding population. So this would be called a founder event. So basically, our two M&Ms, let's say they went to an island, they then beget and populate that island and make this same number of M&Ms. That is considered a founding event. So, bottleneck effect. Change in the gene pool occurs if there's a dramatic reduction of population size. Individuals are killed kind of randomly. And so the alleles in the survivors may not, so a certain amount of allelic diversity may be lost. The alleles in the survivors may not be representative of the original population. Then we have the founder effect. genetic drift in some new small colonies. A few individuals leave their population and establish a new isolated place. This also could be a survivor's effect if if they did not go to a new area, but rather everyone else died. Yeah, and again, the point is when something like this occurs, you lose genetic diversity. Now, gene flow. So, gene flow is going to go and refer to individuals who move into and out of a population and bringing new genes and exporting genes to that population. This can as long as genes are flowing between two populations, in general, we would classify them as the same species, depending on whether you want to get a publication out or not. A secession of gene flow, however, is going to go and help trigger a speciation event. The two populations can start to specialize for different things if they aren't sharing the same gene pool. Now, a species, there are many definitions for species. Historically, we use the morphological definition, which was if they look alike, they probably are alike. Then there is the biological expression, which is if they can produce fertile offspring, then can be classified as different species. Unfortunately, we do nowadays we tend to use the biological definition, but whenever you make a publication and you're trying to make a point, people will go and use different definitions in order to facilitate their paper. So it is something to be important to be it's important to be aware of. But this here is the biological concept of species. Speciation can occur when two populations become separated, and as they slowly accumulate genetic recombination and mutations, they start to no longer become capable of successful interbreeding. So that could be because of genetic changes. Some people would include behavioral changes, but as a whole, it just means that genes are no longer flowing between these two populations anymore. Mutations, changes in the DNA of a gene that can produce new alleles, again, induced by damage. When they are passed on in the gametes, that is going to cause an immediate change in the gene pool because all of a sudden we have something new, and that's going to go and be dictating the creation of proteins inside of our new organism, right? So all of this, we figured this out in the mid 1800s. Research done by a couple different people, Charles Darwin, and I think his name was something Wallace. But they both published books at about the same time. They were both working with each other on each other's books. They both fully credited each other in their work. But for some reason, people only remember Charles Darwin, even though he was part of a two-man team. Anyway, the point is, Darwin and Wallace identified a couple different facts around natural selection and evolution that kind of became foundational for our modern theory around it. One of them is that species are not specially created and they are not unchanging forms. So before this point, it was assumed that creation was perfect. All animals were made in their present form by God 4,000 years before this, and they hadn't been changing over time because why would the divine create a unperfect entity, right? But Charles Darwin was able to go and find physical evidence and records that showed that organisms were changing over time. Modern species are descendants of ancestral species. That was another big development because that also said species can change over time, but it also identified species that no longer exist, species being wiped off the face of the earth, which is something that people did not think was possible before this point. Whenever they would go and find, say, dinosaur bones, it would be assumed that they were dragon bones or whale bones or giant bones. There would be an explanation, but it would not presume that it was, oh, animals on Earth used to be different. Now, in natural selection, individual variation exists within a species, and some of this variation is going to be inherited. This is going to go and leave some offsprings more successful than other offsprings. Inherited characteristics makes them better suited to their local environment. Again, it's important to remember, natural selection does not have a will. It is a statistical phenomenon. And it just means that the individuals who reproduce more are going to be more well represented in the population. And since genetic data can be passed down between generations, if you're really good at surviving, natural selection is going to select for your line. Now, evolutionary change, this is the slow accumulation of positive physical traits through genetics between generations. These will slowly become more and more common within the population as those individuals are going to go and be better fitted for that environment. Now, fitness. Number of reproductively viable offsprings among individuals. This is just our individuals with greater fitness have more of their genes represented in future generations. Basically, what this means is that so as you accumulate positive traits, you're more likely to survive. You're more likely for your offspring to survive to breeding age. Given that over time, we call this gradual transition to a better mode for the environment, the accumulation of fitness. Individuals have static variable fitness. You can only change fitness between generations because when you are passing on these mutations, very often they are not going to be present in you. And with allelic reshuffling, you're going to see new emergent traits that may have never existed in the population at all, without even having to add any new genetic material. Now, adaptation through natural selection. Basically, over time of this happening, as the species settle in and unfit individuals are killed off and fit lines continue to perpetuate themselves, the population is slowly going to be better suited to their environment. It's important to note that all environments are kind of in a state of change most of the time, and they will need to constantly be adapting as their environment changes around them between the generations. So it's important to note, natural selection does not lead to perfect organisms. It leads to a good enough organism. Our evolutionary strategy as a species is for you to reproduce as much as possible before the age of 35, at which point, within the next 5 years, you should be killed while hunting so that you no longer compete for resources with any of your adult children. That's not ideal for the individual. It doesn't sound like a perfect organism, but it's what worked with the evolutionary history of our species. Does not lead to a lot of personal fulfillment. I personally would like to live past 35, but it is what we evolved to do because individuals who were living this life cycle had more offspring survive to reproduce than individuals who weren't. That does not mean that they are better individuals. It does not lead to mean they are perfect organisms, but they are better suited to the environment in which that behavior manifested. Now, natural selection, it's important to note it can act on available variation only. You're not going to go and just pop a duck wing out of nowhere. You have to work with the parts you have. And because of that as well, you can't make any big changes quickly. Existing structures need to be modified. Again, you can't have a duck foot just appear out of nowhere. So it's important to emphasize, no organism is going to be perfectly suited for every niche in its environment. There are many, many different niches to make an ecosystem. And all of them are going to require very different strategies to exploit. Because of this, natural selection might go and be pulling an organism in two opposite ways as well. Like having a trait might help with reproduction, but it might also lead to less frequent reproductive cycles. So that's in part why all this takes a very long time. You have to think of every single variable that is occurring inside of someone's life, inside of an organism, what they are facing. And then you have to go and figure out how that be reflected in the genetics. And then again, you have to go and leave that subject to random change. So here's a little video that kind of talks about the scale of evolutionary change a little bit.
Such mutations in sex cells are heritable and can be passed on to future generations. Mutations are fairly rare. Still, mutation is critical to evolution because it provides the only means for creating new genetic information. A point mutation is a nucleotide sequence error often resulting from incorrect insertion of a single base during DNA replication. Mutations can also be more dramatic. For example, a section of a chromosome may be duplicated, resulting in a longer chromosome containing additional DNA. Likewise, a section of a chromosome may be deleted, resulting in a loss of DNA and a chromosome. Most mutations that arise do nothing or are harmful to an organism. However, a few mutations occur that are adaptive and contribute new genetic information to a population. Gene flow is the movement of alleles between populations. This movement occurs when individuals from one population migrate into the territory of another population with different gene pools. Plants called California tarweeds are found throughout California. Although divided into many local groups, the species forms a single population. Gene flow occurs between the groups, so no group has a gene pool separate from the rest. At some time in the past, one or a few of these tarweeds somehow got to the Hawaiian Islands over 3,900 kilometers away, possibly as a seed stuck to a bird's foot or feathers. Once in Hawaii, the plant reproduced and formed a new population. The great distance separating Hawaii and North America prevented most plants or seeds from traveling from one to the other, making gene flow between the populations non-existent. Therefore, the gene pools of these groups were reproductively isolated. Evolution of the native tarweed and Hawaiian tarweed proceeded separately. As new mutations arose among the Hawaiian tarweeds, those alleles that helped them to adapt to and survive in their new home were selected for. Eventually, new species of plants found only in Hawaii, including the Hawaiian silver sword, arose from the ancestral population derived from the California tarweed. Genetic drift is a change in allele frequencies that can occur by chance in a small population. Consider a hypothetical population of 10,000 penguins in which one individual in 10 carries a given allele. A natural disaster strikes and the population loses half its members, including 550 individuals who carry the allele. The frequency of the allele in the population thus drops from 10% to 9%, but this is a small effect on allele frequency, and no alleles are lost. Now consider a population of 10 with the same allele frequency of 1 in 10. There is now but a single carrier of the allele. If the population likewise loses half its members, and the one member of the population who carried the allele is not a survivor, the frequency of the allele in the population drops from 10% to 0. This allele can now only be replaced by mutation, unlikely, or by migration from another population. An example of genetic drift, the chance alteration of allele frequencies in a population, with such alterations having the greatest impact on small populations. Sexual selection occurs when some members of a population mate more often than other members. In practice, mating based on phenotype, which is any observable trait in an organism, including differences in appearance and behavior. It is the behavior of strutting and chest swelling in a male stage grouse that causes the succession of female grouse to mate with it rather than nearby competitors. It's easy to see that if one male mates 4 times as much as the average male of his generation, his allele stands to increase proportionately in the next generation. Differential mating success among members of one sex in a species often is based on choices made by members of the opposite sex in that species. Some individuals may have
This phenomenon, by the way, is known as intrasexual competition. Intersexual competition, which is sex between the genders. Intrasexual competition with an A is going to refer to like males lecking or like basically fighting each other to try to accumulate females.
survival than other members of their population. Individuals more likely to survive long enough to reproduce are more likely to pass on their traits. Thus, the traits that aid in survival are the ones most likely to be passed on to the next generation. This phenomenon is known as natural selection. Natural selection is the only agent of microevolution that consistently acts to adapt populations of organisms to their environment. Because of this fact, natural selection is generally regarded as the most powerful force in evolution. Sometimes evolution through natural selection can be observed over just a few years. Such an example was provided when a drought occurred in 1977 on Baffin's Major, one of the Galapagos Islands. A large part of the island's population of the finch size of mortals died during this drought. In 1978, it was observed that the average peak depth of the finches that survived the drought was greater than the average peak depth that had been measured in the survey conducted before the drought in 1976. Individuals with the larger beaks were better able to crack open the large tuft seeds that were available during the drought. Finches with smaller beaks starved during the drought for lack of an accessible food source. The offspring of the survivors also were observed to have a larger average beak size than the population before the drought. Thus, the population had evolved through natural selection over the course of just a few short years.
Yeah, and this Darwin's beak experiment is super famous. It was done by a couple named the Grants. They were studying Darwin's finches from like 1970 to now, basically. Like they're still working out there. Now, macroevolution, this is going to be large-scale evolutionary change over very long periods of time. You can think of it as accumulation of microevolution events. So Linnaeus, scientist who developed the Latin binomial scheme for classifying organisms in the 1700s, he's got figured out genus names followed by the specific species epithet. He created the taxonomic system that we use to this day for organizing organisms. The Linnaeus system is basically takes very broad categories and gets gradually more specific with them. Domain was not added by Linnaeus. It was actually added later by us. Alrighty, so if you look here, the lowest level of organization that Linnaeus defined is species. We have the biological species concept at this time. He was working on the morphological species concept because they didn't have genetic evidence. Then there you have genus. These are closely related animals that cannot interbreed. Then family, order, class, phylum, and kingdom. Then above here, we would have domain. We pretty much developed domain because we realized that we realized some things that we did not know were alive were actually alive. All right, so let's look here. First, you start at the top with domain. This is going to be things like eukarya, prokarya. What type of cell are you? Then you have kingdom. This is going to be animals, plants, fungi, bacteria, archaea, just the different large categorical groups. Then within here, you'll have like chordates. You'll have chordates. You'll have the sharks. You'll have amphibians, mammalia, stuff like that. Or I'm sorry. Up here you have chordates. These are all the spined individuals. But this is, again, a broad category. All these animals have a spine. That is their common trait, so they are all chordates. Then you have subphylum vertebra. Vertebra is going to only include things that have like a rigid spine or a bony spine. These guys don't have a bony spine. They don't have a vertebra. Then you have mammals, bird, lactate, primates. That includes us, two nipples, by semi-arboreal family, Homininidae. This is going to be the great apes. Then you have genus. That's going to include humans and the close human relatives, things like Neanderthals, Homo erectus, Homo ergaster, our closest relatives. Then finally, Homo sapiens, the modern human species. It is important to note, occasionally, additional categories will be inserted into this depending on the publication. You might have subspecies. You might have infraorder. You might have infrakingdom or subkingdom. All of these are just going to go and be reworking this system that is 300 years old to work for the publication that they are trying to publish now. Uh, Taxonomy is a very, very messy thing. It's not something I really like very much, but it is the system by which we organize all life on Earth. And it's a system that we have agreed to internationally, which is even more important. Just getting everyone to use the same standard set of measurements is a huge achievement. Now, when we are examining genetic histories, lineages of species, we develop what's known as a phylogenetic tree. So, phylogenetic tree is going to be a branching diagram that, in theory, should give you a rough idea of how closely related species are. So let's go and have a look at a very primitive one and the methodology they used to go and make this. So here you have sharks, frogs, and humans. Each of these are going to have two paired appendages. Only frogs and humans are going to have digits, and only humans are going to have hair. So we now have three traits, which we can go and see as like divergent periods in the evolutionary history of these animals. So first divergent period, development of two paired appendages. Sharks are out. They did not develop two paired appendages. And so our lineage, the tetrapods, continued on. After a while, digits evolved out of fins. Pretty darn cool. No digits here on a shark, which is how we know the shark was not here. Oh, sorry. Two paired appendages. Everybody's two pairs of appendages. Sharks do not have digits, so it's likely that the sharks split off before digits developed. Then you have the frog. Frogs do not have hair. So it's likely that frogs broke off after the digits developed, but before hair developed in this phylogenetic tree. As we get further up, this is going to be further forward in time. So you can kind of see this as like a timeline to a degree, like this horizontal line right here. It's an inverted family tree, is another way to look at it. Now, evidence of evolution comes from several sources. The fossil records one, we just keep finding fossils that look like steadily transitioning forms between two other fossils. And we just kind of realized that there was like a spectrum of fossils that slowly changed over time. They showed that the species were changing over time. Geographic distribution, also known as biogeography. You can find a lot of different species that are pretty closely related to each other on different continents than each other. You can go and find plant communities that that are only present in like South America and Australia. You can go and find fossil records for animals that would not have been able to survive in the current environment that's in different regions. Basically, it can go and help tell the story, give the narrative, and kind of guide your explanation to go and have a better understanding of geographic distribution and the conditions that the fossils were subject to while they lived there. And then we have molecular biology. Now we're getting to a point where we can use comparative molecular biology. So just comparing the genetic information of different individuals to each other. And then finally, you have comparative anatomy and embryology. This is how Linnaeus was organizing all this. This is how taxonomy worked until about the 80s when we got really good at when we started to get good at genetic stuff and genetic analysis. Comparative anatomy and embryology is something you should be familiar with, and it can be a useful tool, but it is not something that you want to make a decision using because we have better tools now for specifically designing taxonomies. Now, fossils. Preserved remnants and impressions of past organisms provide strong evidence of evolution. However, it is a biased sampling of past life. Larger animals are disproportionately represented in fossilization. It's easier for a larger bone to be fossilized. And animals with thicker and heavier bones are disproportionately represented as well. So we're missing a lot of microfauna. We're missing soft tissue. We're missing animal behavior. We're missing genetic evidence. We're missing a lot of the stuff that would allow us to verify a lot of the claims that we're making from the fossil record. Now, fossilization. Hard calcified tissues like bones, teeth, and shells become impregnated with minerals. usually from surrounding water and sediment in an anaerobic environment. Eventually, this is going to get pressed down and compressed into like a sedimentary rock. And at that point, erosion is eventually going, or excavation is going to eventually expose the sedimentary rock that this imprint is going, that these samples are going to be lodged in. And then that can be mined or that can be collected or whatever you end up doing. Here are a couple different examples of fossils. Here is a spider that got fossilized inside of a chunk of amber resin. And here is a fish that died in the middle of eating a fish, and most likely choked to death, that then had an impression of its body and bones saved in the sedimentary rock. And this is how that process works. Organism dies underwater, sediment accumulates on top of them. The hard parts of the organism become impregnated with minerals, usually calcium. Then eventually uplifture and exposure expose the fossil to be grabbed for a museum. Now, the fossils of extinct organisms that we found often show similarities to and pretty extreme differences from living species. We have also been able to find a lot of ancient organisms that we can directly link to modern species because of shared or ancestral traits. So we'd have something. So we have a whale evolution is pretty well documented. We know that they came to land, then they returned to the water, and now we have whales, and there are transition fossils from every single stage of that. Now, biogeography is the study of the geographic distribution of organisms. Biogeography is often going to reflect the evolutionary history of different organisms and different relationships. And it's also going to go and be working on the principle that animals are going to be more closely related to the geographically near animals than they are to ones that are very, very far away, which makes sense. I mean, think about the Darwin's finch model. We could not, or the tarweed model. We could not get more tarweed to Hawaii. So it started to speciate. So here's a hyper-specialized filter feeder, a bowhead whale. This is one of their ancestors, still fully aquatic, but is a toothed whale. Another one of their ancestors. This was more like a crocodile, both aquatic and terrestrial. And then one of their distant ancestors, semi-aquatic, spent a lot of time on land. We also found certain structures in their feet that they have in common with artiodactyls, which is going to include hippos, cows, deer, pronghorn antelope, all the double-hoofed mammals. It turns out that whales are actually very closely related to them. And one of the ways that we figured this out, even though we didn't have genetic evidence until pretty recently, is that there are a lot of common retained structures in their bodies that don't actually have any utility. Nature does not make waste. So if there's a structure that is useless, you can reckon that it's retained from a prior time when it had a use. And if two species have a useless structure, it's statistically likely that they came from a common ancestor. Now, geographic distribution. New distributions of organisms occurs by two basic mechanisms. There's dispersal, when organisms move to a new area. Areas occupied by organisms move or are subdivided. So what's this basically saying is, so dispersal is where organisms move to a new area and colonize it. And then after that, that's going to lead to a new set of selective pressures. A new animal has emerged in the ecosystem. So let's look here. So marsupials most likely originated in the northern hemisphere, some around 125 million years ago, most likely somewhere around where China, although at the time could have also been North America. Like this whole spread, there was a lot of gene flow between these two. From there, marsupials arrived in South America during the South America, Antarctica, and Australia following these land bridges. At this point, most of this region of Antarctica was temperate rainforest. Over time, Australia drifted away from Antarctica and started going butt into Indonesia, which isolated this population of marsupials from the rest of the world. And because gene flow was no longer occurring, they started to speciate. They were specializing through the random action of selective pressures towards being more appropriate for their environment, while their relatives in Antarctica, South America, and North America were having to do the same. The only marsupials present in the New World to this day are the opossums, because every other marsupial was outcompeted by every other animal. Now, comparative molecular biology. So point mutations in DNA in most animals are going to accumulate at a pretty consistent rate. Exposure to sunlight's pretty universal as inducing mutations, but these guys aren't going to be exposed to a huge amount of carcinogenics. So we can pretty consistently figure out the rate that mutations are going to occur in a species. Humans are a bit weird. We mess with a lot of stuff that screws with our DNA. We accumulate mutations kind of fast because we're being exposed to hazardous chemicals more often. Now, because of the molecular clock, we can actually figure out when two species diverged based off of how many mutations they've accumulated since that point that were not accumulated by the other species. And because this happens at a consistent rate, we can actually get a time estimate for when they diverged. So this brings us to two topics. These are going to be comparative anatomy, also known as homologous, and I can't remember the word. It shows up later in the presentation. Homologous and analogous structures. So homologous structures are structures that are similar, most likely because they came from common ancestry. This is going to, in particular, include structures that do not have a purpose. Form sometimes follows function. You might have two structures that are unrelated that look similar, but if a structure is useless and it's present in a lot of organisms, it's most likely in the process of evolving away, and it's most likely retained from a common ancestor. As you can see here, every one of these animals uses the same bones of the limbs in similar positions, just adapted for different purposes. These are all the same bones. They've just been rearranged. Now, another aspect of this is common embryological origins. At different stages in development, if two organisms look similar, the more similar they look later into development, the more closely related they likely are. So let's have a look here. Here we have a fish, a chicken, and a human. I would say chicken and human look pretty comparable in these first two panels. Third panel, they've obviously differentiated. Fish, second panel, you can pretty much tell what it's going to be. That would imply chickens and humans are more closely related than humans and fish, which is true. Now, now we have this little video that's going to go and talk about some of the biogeographical history of the Earth.
During Earth's history, the tectonic plates that make up the surface layer of Earth have moved with respect to one another, causing the continents to spread apart or collide. About 250 million years ago, a single supercontinent stretched from pole to pole. As a result of continental drift, the landmass began to break apart, creating continents that take the shape of those we know today. The movement of the continents has had an enormous impact on the distribution of animals around the globe. Let's take the example of marsupials, which are mammal species such as kangaroos and koalas, in which the female often has a pouch. Most species of marsupials today live in Australia and nearby islands, with many fewer in Central and South America, and just one native species in North America. The geographic distribution of organisms is called their biogeography. How did the marsupials become distributed in this pattern across the globe? Let's look again at the movement of continents. One possibility, based on a body of fossil evidence, is that marsupials evolved in North America more than 100 million years ago. Then about 65 million years ago, some dispersed southward into South America, then to Antarctica, and later to Australia, to which Antarctica was attached at the time. Subsequently, Australia drifted away from the other continents, isolating its resident marsupials from similar influxes of species. The marsupials of Australia vastly diversified. Those in South America did as well. In contrast, in North America, placental mammals began to diversify and outcompete the marsupial mammals on the continent, leading the resident marsupials to extinction. About 3 million years ago, the Isthmus of Panama formed, connecting the Americas by a land bridge. The connection allowed a migration of animals both up and down the Americas. The result was a massive.
This was known as the Great American Interchange. It's also how cats got to South America, and how armadillos got to North America. Pretty cool.
Loss of marsupials from South America, as they were outcompeted by the placental mammals from the north. The single North American marsupial species, the Virginia opossum, arrived from South America by this route. In contrast, Australia continued to remain isolated, allowing their resident marsupials to evolve in isolation.
Yeah. So the marsupial reproductive strategy is significantly less successful than the placental reproductive strategy. In any niche where a marsupial is competing directly with a placental mammal, almost always the marsupial is going to go and be pushed out of that niche. That is in part why possums have thrived, because they have several conditions that make them very different from most other mammals, period, on Earth. They have a lower body temperature, which makes them immune to rabies, and it also makes them immune to Lyme disease. So because of that, they could specialize in eating things that nothing else wanted to eat and make their own new niche. Most of the possums fit somewhere in that area for the New World possums. In Australia, you see a lot more diversity because they could fill the niches that otherwise would have been filled by placental mammals. Now, convergent evolution is going to be based off of one biological principle. Form is going to follow function. So if you have a similar job to do, oftentimes you need a similar tool. If you have two individuals that have a similar physical structure that are not related to each other, that are fulfilling a similar, because it is most likely because they are doing the same job. And we would call those two structures that are similar because of we would say they are similar because of convergent evolution, and we would call them analogous structures. Structures that follow function and are not retained from a common ancestor. Now, humans, we trace our roots back to the first primates. We most likely arose from a series of insect-eating mammals that lived in trees, kind of like a tree shrew or a squirrel. Over time, a lot of our adaptations are associated with being an arboreal squirrel-like insect eater. We have super sensitive rotational shoulder joints, sensitive pads on the end of our digits. We have nails, and we have grasping feet and hands. All of that is associated with insect capture. If you look at like the tiny hand of like a shrew, they kind of look like a human hand. It's really weird. Now, eventually we stayed arboreal, but we started to move to different modes of motion. One of the most common being brachiation, which is where you slowly swing yourself underneath something. It's the way a kid uses a monkey bar. And we started to shift a little bit less away from insect selection and towards fruit selection. So fruit selection and arboreal lifestyles, they're going to go and trigger larger brains, and they're going to trigger forward-facing eyes. If you are arboreal, depth perception is super important. Forward-facing eyes give you depth perception. If you have a large brain, you can remember all the different fruits and which ones are poisonous and which ones aren't, and all the different bugs and which ones are poisonous and which ones aren't. And you can remember where all those are and at what time of the year. You're not going to forget just because it's been 6 months since the last harvest. You'll remember you had a good meal there 6 months before. And they also started selecting for smaller litter size. I personally think that's just because being a frugivore requires you to go and be kind of mobile. You have to travel a lot. And an arboreal species cannot travel with more than 1 or 2 babies. Now, order primates. Within the primates, the first suborder that we can look at are going to be like the primitive primates. These are going to be the lemurs, the lorises, and the podos. They have a lot of our ancestral features. So this is kind of what we looked like when we were arboreal. Probably physically smaller, never as rangy as the Poto, as the Slender Loris, because it has a few other adaptations that are associated with like nocturnality and things like that. But yeah, we definitely have ancestors that look like a ring-tailed lemur or a Poto. Now, moving on to another suborder, the monkeys, the apes, and the humans. This is going to go and include the family Hominidae, which includes both apes and humans. Within Hominidae, Hominidae will also refer to these as the hominids. And this is going to go and include, all the monkeys, all the apes, here's a cute Western gorilla, a little Bornean, Chacma baboons, all that. Now, within this family, here is our phylogenetic tree for all of these taxa. Here you can see 65 million years ago, we had a really early diversion with the lemurs, the podos, and the other primitive primates. Then later, we had a diversion. Then later, the New World monkeys became separated from the Old World monkey population, most likely through a land bridge. Most likely through a colonization event, a founding event. Then the Old World monkeys started to differentiate. Then after that, the gibbons became hyper specialized for arborality, and they started to lose their tail, and they began to differentiate. Now our lineage started to go and specialize into the Asian apes, so then the orangutans differentiated, and the African apes. Within the African ape lineage, about 6 million years ago, we diverged from chimpanzees. That was like our last common ancestor, in part because we started to go and move towards more living in more savanna mosaic environments, while chimpanzees preferred to stay in pure forest environments. Now, let's do some comparisons between human and chimp skeletal anatomy based off of that 6 million year ago diversion. This is about when we shifted from being primarily from using trees pretty much constantly, being aggressively semi-arboreal, and then starting to transition to a terrestrial lifestyle. So, human skeletal anatomy is going to go and reflect this. Bipedalism, very unique trait, kind of novel as well. You don't see it very often. It's going to go and be depending on having a S-shaped spine, a large patella, arms that are shorter than the legs, and a stiff foot to support the body. The less opposable the toes are, the more they are able to make a flat pad with the ground. So in all these structures, you can see chimpanzees, their big toes opposable. It's pointed towards the other toes. Hands, they are robust for carrying weight and enabling arborality. Ours are fine for refined motions, things like writing and making detailed tools. Yeah, and then from there, it's just a series of adaptations that are going to go and enable the lifestyle that we needed to engage in when we were going into the savannah. Stuff that's going to go and let us walk all day, and stuff that's going to let us problem solve. Now, common misconception about evolution. Humans are not descendant from chimpanzees. We are descendant from a common species that beget both chimpanzees and us. So we do not come from them. We just have a common ancestor. We diverged about 6 million years ago. So it's important to note as well that humans did not have a real linear evolution. Our current DNA shows hybridization with a couple other species of humans, including the Denisovians and the Neanderthal, and they occupied all different areas. All of them are extinct in the modern era. Some of them were driven extinct in part by us. Others just died out on their own. But the point is, we've been interacting with other human species for a long time in our evolutionary history. Now, different parts of the human body evolved at the same rate. Different traits evolve at different times and rates. This is known as mosaic evolution. Like, bipedalism might have happened quickly, but something like, say, shortening the jaw and flattening the face, that might have taken longer. Reducing sexual dimorphism and body size, that could have happened really quickly, actually. Increasing brain size with tool use, that would have been super, super slow. And language and behavioral complexity, that's going to be directly correlated to brain size. So, Now, one of our oldest hominins. So hominids with a D, that's going to include the apes and the monkeys. Hominins, those are going to be our immediate ancestors, the ones that are going to be intergenous. Intergenous or directly begetting our genus or closely related to our genus. So the Australopithecines beget our lineage. The oldest hominin remains we found are all Australopithecines. The oldest one we found is from Ardipithecus. It's about 4.4 million years old, and it did have some. It was a species that had facultative bipedalism, which meant that it was bipedal as its default gait. We're pretty sure that this then begat Australopithecus, which is like Lucy, like the real classic ones. So species within the genus Australopithecus, or at least some of these recent. So we have Anamensis, occurred about 4 million years ago. Then we have Afarensis. This is going to be Lucy, about 3.2 million years old. We are reasonably sure that some of our progenitors came out of the Afarensis population, because this was more focused on the regions of Africa where our ancestors came from. And also they were adapting for climates and had emergent traits that we saw in our ancestors. So the most recent one we found is A. africanus. That one's only about 3 million years old. Here's a reconstruction of Lucy. I got to see the original bones at the Smithsonian, actually. It was a really cool experience. But as you can see, we are kind of we have the basic human body plan, non-opposable toes, longer legs than arms, straight spine, all that stuff. But there are still a lot of ancestral traits from those ape ancestors, the ones that were like chimpanzees. Now, moving into our actual genus, Homo, the first species we found was Homo habilis. So Homo habilis, also known as the handyman, is about 2.5 million years old. It was the first member of the modern genus of humans. Had a much larger brain, most likely engaged in pretty advanced tool use, and we're pretty sure they were capable of speech. Now, so Homo habilis is a bit of a confusing species because it is either the same species as Homo ergaster, Homo erectus, and Homo heidelbergensis, or all of these are just Homo erectus, or they are all different species. We do not have a lot of genetic evidence from these samples, and so we haven't been able to conclusively say anything yet, but it is likely within our modern definition that all of these species might be subpopulations of Homo erectus. Within the biological concept of species, these are most likely all one species. But because we don't have genetic evidence, we are using the morphological concept of species. And given that, says Homo habilis was its own species 2.5 million years ago. And then Homo ergaster showed up 1.9 million years ago. And in general is considered the population that would begat Homo erectus. Homo erectus occurred about 1.6 million years ago, and it's also the first hominin to migrate out of Africa. We are almost certain to use fire. We know it made boats. We are reasonably sure they made clothing. So the Homo erectus population, they spread throughout most of the old world, Asia, Southeast Asia, Indonesia, Australia, the Middle East, Europe. And we are reasonably sure that they caused mass extinctions wherever they went. They had a similar hunting strategy to what the ancestral human used, like modern humans evolved to use. And that hunting strategy is really good at driving shit extinct. So we think that this caused a first wave extinction about 1.6 million years ago. Then Homo ergaster, Homo erectus, Homo habilis, Homo heidelbergensis is going to be our next one on our list. This is this is this is the sub-Saharan African population of Homo erectus. These are descendant from Homo erectus that never left Africa. About 800,000 to 130,000 years ago. That's closer now to like 230. Uh, we are pretty sure that that this population diverged into two populations, one that became Homo sapiens and one that became Homo neanderthalis. Homo neanderthalis dispersed up north to Europe first. We stayed behind. Now, Homo sapiens or thinking man, oldest fossil evidence for modern humans comes from Africa about 130,000 years ago. We have now found ones that are about 200,000 to 300,000 years ago, which is kind of impressive. The main characteristics are a large brain and a flat forehead. That's the best way to ID the skulls as being different. So, characteristics of Homo sapiens. Absence or very small brow ridge. Modern specimens have no brow ridges. Very old specimens have brow ridges. A prominent chin, the presence of a chin. Most other apes do not have chins. And an extremely slender body form. We have very long, narrow limbs compared to the other apes. And pound for pound with height, we're going to go and be way, way lighter. Now, Homo neanderthalensis went up to Europe and Asia 200,000 to 30,000 years ago. They had a wide variety of lifestyles. We know they made shelters. We know they would fish. We know they hunted in many different ways. The ones that lived in caves were the best preserved in the fossil record because of the conditions of caves. But for a long time, there was a misconception that ancient humans did not make shelters and only lived in caves because we had no physical evidence of structures. Since then, we found physical evidence of structures, and so that has been rethought. Homo neanderthalensis most likely would have been about as intelligent as a standard human, and it is very likely that we just absorbed them into our population, given that most Europeans and Asians do have a pretty high percentage of Neanderthal DNA. It's probable that they just kind of were absorbed and integrated. For a long time, we thought we killed them off. Other time, or hunted them to extinction. But to be honest, reality is usually more boring than that, right? So, the archaic form of our species is known as Homo sapiens robustus. So or Cro-Magnon, and this is going to go and be the caveman. These are going to be our ancestors before 10,000 years ago. After 10,000 years ago, towards the modern era, our bone structure started to become way more delicate, along with our musculature, in part because we didn't have a selective pressure to be good at large game hunting. And because of that, we kind of started to speciate a little bit, but not enough to be considered a different species. We are still the same species as the Cro-Magnons, but we are now classified as Homo sapiens sapiens, while they are Homo sapiens robustus. And what does that mean? That means we are different subspecies. Notable populations that still could have gene flow between them, but are considered the same species, but do not have gene flow between them for whatever reason, and are different enough that it is worth identifying them as such. Yeah, so the Neanderthal, they had a lot of adaptations for cold climates, thicker limbs, a lot of modifications to the respiratory tract, including the sinuses, short, flat nose, and they also produced a lot more body heat than us. Now, Cro-Magnons, they looked like us, just a little bit heavier, stronger musculature, thicker brow ridges, thicker bones as well. Just in general, but if you saw either of these walking down the street, you would not doubt that that's just a guy, you know? All right, so here is just a little review of some of our milestones in human evolution. Here's the lineage history, and this is the actual phylogenetic tree. Again, we do not know if any of these species directly begat any of the later species. All we know for certain is that these are all closely related, and this is when they occurred in the fossil record. You see this little, yeah, Yeah, so we did not actually find a skull for Australopithecus anamensis. We only found finger samples, if I recall rightly. Now, there are two hypotheses. This is the obsolete hypothesis. This has been disproven hundreds of times, but we still talk about it because it comes up every once in a while. The multi-regional hypothesis claims that Homo sapiens evolved independently in Europe, Asia, Africa, and Australia. It claims that there were just four different speciation events that beget the different major races. This is a perpetuation of social Darwinism and scientific racism from the 1800s. It isn't a valid theory, and in general, people trying to engage in this theory are engaging in bad faith. They just, quite frankly, they're usually not happy by the fact that the root of humanity came out of Africa, and they're trying to find a way to complain about it. Second hypothesis is the out of Africa hypothesis. This is supported by fossil and genetic evidence going back since like the 50s. So, out of Africa hypothesis is that we have a single point of origin in sub-Saharan Eastern Africa. Later, Homo sapiens evolved from those species and dispersed first to the Middle East, then to Europe, then to South Asia, and then to Australia, and then from there to the rest of the world. Alrighty, folks. So this should have given you a bit of a ballpark understanding of like the early Earth and how conditions lead to the formation of organic molecules, genetic material, prokaryotic cells, and eukaryotic cells. The differentiation between the processes of microevolution and macroevolution using these terms: population, gene pool, mutation, genetic drift, bottleneck event, founder event, gene flow, and species. And you should have an understanding of human evolution from the primates to Homo sapiens, noting distinguishing characteristics of the early hominids. Try to get to a point where you can go and refute some of the common misconceptions that we talked about, and you're golden. Alrighty, folks, that does it for that chapter. I'm going to go and post this, and I'm going to move on to the next one. Y'all have a good day.